Hi, my name is Mike Ford, and I'm a scientist at the Northwest Fisheries Science Center in Seattle, Washington. I'm going to be talking to you today about killer whale paternity and inbreeding in the southern resident population. I'd like to acknowledge this is a collaborative effort between a number of folks at NOAA Fisheries, including Kim Parsons, Brad Hansen, Candace Emmons, Luna Park, Eric Ward, and Marty Cardos. And this is also in collaboration with Ken Balcom and his colleagues at the Center for Whale Research. So when you think about what causes extinction, it can be helpful to divide it into two basic types of factors. There are environmental factors, which influence the population through things like lack of food, or disturbance from vessels and noise, or contaminants in the environment. And then there are also population factors, and many of these are influenced by population size. So just being a small population is inherently risky, and it can also lead to things like inbreeding, randomly skewed sex ratios that can have an influence on the population, and a lack of genetic diversity needed to adapt to changing environmental conditions. So these environmental factors and these population factors interact. You can imagine that a population could be knocked down through environmental factors, and then some of these small population factors could take over and lead to further problems. And this has been termed an extinction vortex in the way that these things could interact together, something that we obviously would like to avoid if possible for the southern resident killer whale population. So I'm going to be talking today a lot about breeding structure, and so it's worth spending a little bit of time asking how breeding structure affects extinction risk. And breeding structure is important because it could have effects on genetic diversity. For example, if whales from different populations interbreed, that can increase diversity, and if they only breed within a population, that would decrease diversity. It can also affect inbreeding. So if relatives tend to interbreed, the population would be considered more inbred. And if they tend not to interbreed, then that would be a good thing and might result in more outbreeding. And then we're also interested in breeding structure because it could influence the opportunities for mating. So for example, if whales in the population have certain customs that limit certain types of mating, that could also potentially influence the rate of population growth. So we use genetics to study breeding systems, and this is in part because there are often big differences between the observed social breeding system and the actual genetic breeding system. And of course, it's the genetic breeding system that influences all these things like inbreeding and patterns of genetic diversity. So one kind of dramatic example of this is in the case of passerine birds, which observationally often are seen to be monogamous and were thought to be monogamous as late as the 1970s. But in fact, about 86% of the species studied genetically turn out to be polygamous. So in this case, the observed breeding system and the actual breeding system could be quite different. And of course, this could occur in a number of species as well. And one of the kind of risks, for example, of going to companies like 23andMe or other genetic companies for looking at human ancestry is finding out that your own personal history might be different than what you expected it to be. So in the case of the killer whales, the maternal pedigree in the population is well known through observational studies, and these have been conducted by folks at the Center for Whale Research and up in Canada and a number of other organizations for a period of nearly 50 years. And the reason why this works so well for the maternal relationships is that the offspring swim with their mom, certainly when they're young and nursing, and often for much of their lives. So by watching who is swimming with whom, you can determine who the moms and their offspring are. But as you can imagine, this doesn't do a lot of good for the dads because the dads typically are unknown through this sort of observational study. So that's where the genetics comes in. And here we do a basic sort of paternity analysis, very similar to what you would do in, say, a human paternity court case. And typically we have a mother whale and some of her known offspring. And if we have a genetic sample from mom and offspring and all of the potential fathers, we can use the simple rules of genetics, the fact that one copy of each gene comes from mom and the other copy comes from dad, to figure out which offspring were potentially produced by which father. So you can see in this example here, if we're looking at mom's DNA sequence, this particular part of her genetic code, she has 2 T's that she got from her own parents. And then she passed on one copy of these to each of her offspring. So in this case, each of these offspring got a T from mom. But you can see that they also got a different gene from their dad. And if we go to the dads, we can see that, for example, in this calf, of these 3 potential dads, the top 2 could potentially be the father of this calf, because either one of them could have contributed an A. And if we look at this calf over here, we can see that if these are the only 3 dads in the population, it had to have been the dad in the middle, because he's the only one that has a C at this particular gene. So this is the basic idea behind paternity analysis. You typically use many more genes, either dozens or potentially thousands of genes, to do this much more precisely, but this is the basic idea. So in order to study the genes, you first have to collect the DNA, which means collecting tissue samples from the whales. And there's really 2 ways that, that we've done this. One way is to collect a biopsy sample with a small biopsy dart. It collects a little bit of tissue and blubber about the size of a pencil eraser. And then the other way is to collect fecal samples. And this involves following the whales and collecting the poop, basically just with a swimming pool net, and collecting DNA in that way. So let's give an example of this. So here we have K14 and her calf K42, born in 2008. And so here's a known mother-calf pair, and we're interested in knowing who the father of this calf is. So in this particular case, there are about 20 potential fathers, and they're all listed here. I'm not going to go through them all. And nearly all of these fathers were sampled in the case of this particular calf. So we have a good collection of the potential dads. And so let's figure out who the dad is. And in this case, it turns out after doing that analysis, only one of those 20 dads has a genotype, has the genes that are consistent with the calf K42. And so we can be really confident that L41, male L41, is the father of this calf K42. And it turns out L41 is also the calf of K42's brother K36, but he's not the dad of her other brother K26. And in fact, the father of K26 was not among any of the whales in our sample, which isn't too surprising because he's an older whale, and only a few of the potential dads were able to be sampled while they were still alive. So in some cases, we have neither a mother nor a father sampled. So for example, in 2013, neonate was washed up on Dungeness spit. So in this case, we don't have the mom. But we can still go through and do the same sort of process with all the potential moms and all the potential dads. And in this case, we could be quite confident that the mom was J28 and the dad was L41. So here's sort of what the big picture looks like. This is a family tree showing all of the matrilines and all of the patriline's. So the matrilines are shown in the light gray here, and so each one of these is a mom, and it shows her birth date, and then the birth dates of all of her offspring. And the moms are not labeled in this picture, just for sake of clarity. And then the dads are labeled, and it shows the dads and their birth date, and then all of their offspring. And there are a total of 8 dads in this picture, both dads that are sampled, that are labeled, and then some dads that are unsampled, and they're labeled with these stars. And so a couple things are interesting when you look at this. First of all, you can kind of see that there are two males, J1, born around 1950, and L41, born in the early 70s, who were clearly sort of the dominant males, if you will, at least for the group of samples that we had. So you can see that particularly if you focus on this time period after about 1990, more than half of all of the individuals in the family tree are offspring of either L41 or J1. And you can also see that there are a few other whales that have contributed, males that have contributed offspring, including J26 and K26, L57, L79, L78, and L85. But they've only contributed, at least at the time that this tree was produced, a few offspring each. And then if you go back in time, you can kind of take some guesses at males that are not part of our sample, but were sort of fecund males simply based on the half-sib relationship. So for example, this male, star 7, we'll call him, produced quite a large number of offspring born around 1990, including both J26 and K26 as an example. So one of the things that we were interested in doing this study was to ask how inbred the population is. And of all of the offspring we looked at, which was maybe about 80 or so, we found 5 that were highly inbred. And so those are listed here. And so you can see, for example, J42 had a mom, J16, which was known, and dad, J26. And that's a mother-son relationship. So J42 is highly inbred because his parents were highly closely related. Similarly, J46 was the offspring of a father-daughter mating, so also very, very highly inbred. There's a couple K animals, K34 and K33, that are each inbred, but a little less so, either half-siblings or uncle-niece in the case of K33. And then more recently, J52 was the offspring of J26 and J36, which is another half-sib mating. And when you think about it, if you go back, I'm going to go back one slide. Actually, I can't go back one slide, I'm sorry. When you think about it, if you remember that previous slide, the two dominant males, J1 and L41, produced about half of all the offspring born after 1990. So a lot of the matings that are going to occur from 2000 on are potentially going to be amongst individuals who are half-siblings. So one of the reasons why we're interested in studying inbreeding is that it could be a potentially important health marker. So inbreeding, as I think you've probably gathered from this talk already, just means mating between relatives. That could be close relatives, or in some cases more distant relatives. Inbreeding leads to lower genetic diversity, and it also potentially leads to inbreeding depression, which is reduced fitness, basically reduced survival or reduced reproduction of inbred individuals compared to non-inbred individuals. And the reason for this is that all of us, humans and many other species, have lots of deleterious alleles throughout our genome. Alleles, it genes that if you have 2 of them together, it can be lethal or can lead to lower fitness. And mostly that's not a problem, because if you are the offspring of unrelated individuals, you usually only have 1 copy at a particular gene of those deleterious alleles. But if your parents are highly related, the odds of you having 2 copies of a deleterious allele are very high, and that can lead to lower fitness. So in looking at the southern Roseland killer whale population, one of the things we found is that there's no real evidence of inbreeding avoidance. So one might imagine, for example, that maybe the whales have ways of knowing that they're related and would avoid mating with close relatives. And we didn't really see any evidence of that. And you can see that in this figure. The x-axis along this figure is a measure of the degree of relatedness among individuals. And if you're kind of more on the right, you're more related, and if you're more on the left, you're less related. And the black dots just show the actual actual matings that we observed in the population, the degree of relatedness among moms and dads. And then the gray dots is just if you sample the population randomly, what would it look like in terms of pairs of individuals. And you can basically see that the black dots and the gray dots, and it's illustrated in these curves here, are pretty much overlapping. So there's no real evidence that the actual matings are any less inbred than what they would be if the whales were just mating randomly in the population. We do have a little bit of a hint that being less inbred leads to higher survival. So this is again a kind of a graph of some degree of a measure of inbreeding and an estimate of the annual survival rate of the whales in the population. And you can see that basically if you're a little less inbred, you tend to have a slightly higher survival rate. And if you're a little more inbred, you have a slightly lower survival rate. But this gray shading around the line is the confidence interval around that line, and you can see that we're not very confident in this relationship. One of the things that we are currently doing to become more confident in some of these relationships and understand the consequences of inbreeding better is to sequence the entire genomes of all of the southern resident killer whales that we have sampled. So we have about 100 samples from southern resident killer whales, and we're sequencing their entire 3 billion base pair genome each. And then we're doing the same for about 50 individuals from other populations of killer whales throughout the Northeast Pacific in order to compare degree of inbreeding of the southern residents to some of these other populations. We're still in the process of analyzing these data, but I just wanted to show you kind of an example of what this looks like. So in this case, I'm showing a picture of the degree of heterozygosity across 3 different chromosomes for these whales. And it's just a sampling of whales. It doesn't really matter who they are in this particular example. And you can kind of see that for each of these whales, and some more than others, there are segments of their chromosomes where they basically have no diversity at all. And by examining the patterns of how big these segments are and how frequent they are, we can get a really accurate picture of how inbred each individual whale is. And by looking at what genes are contained in those segments where they have no diversity, we can also estimate the consequences of that inbreeding and how likely they are to suffer from low fitness. So, in conclusion, some of the key points from my talk today were that the study showed that the male reproductive success in the southern resident killer whale population is highly skewed, with about 50% of the recent population descended from just 2 males. We found 5 apparent cases of very close inbreeding in the pedigree, including inbreeding between first-order relatives such as parents and offspring. The consequences of that inbreeding for survival and reproduction are still under study. We have some hints that that could be a problem, but we don't really know yet. We found no evidence of inbreeding avoidance, and we found that the population has had a low effective size for at least several generations. So with that, I'll come to a close, but I thank you for listening to me today, and I think later I'll be happy to take any questions.